Okay, so now that we have aseptic technique down, the next experiment 6, was making a pure culture and isolating a colony. So, the way to do this is to do all the steps of the aseptic technique, but instead of putting it on a plate like this, you do it onto a or instead of doing it on a slide, you do it onto a plate. And so right here, I got my plate. I made it out of tape. It took like 20 minutes to do, but I'm proud of it. I know. Okay, so alright, so you do your aseptic technique, but instead of putting it onto your slide, you put it onto your plate. And so the first thing you do is you label it. So you put your name, your date, the bacteria that's going on it, probably some other stuff. Read your lab manual to make sure. Oh wait, I have it right here. Section number and your initials. Okay, so you put all that on, and then you divide it into 4 things like we've been doing. And then so that's all ready. You want to make sure to do the agar side and not the lid, because the lid can be lost or whatever, but the agar will be always attached to the petri dish that you are labeling. So you take your petri dish, and you want to make sure to always keep your lid on there as much as possible. So when you're doing this, you only want to remove the lid when you're actually interacting with the actual agar, instead of just taking it off and then doing the whole process and then putting it back on. You want to take it off, do 1 quadrant, and then put it back on, flame it, blah 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 blah, take it off, do a quadrant, put it back on, and that'll make less contaminants. Some of y'all might be getting lucky and then doing that but not getting any contaminants, but I think if you get a contaminant in your colony isolation, then they take off like a point or 2. So those points are precious. So try to avoid that, and correspond to aseptic technique as much as possible. So, all right, when I'm going about this, you take your thing and you streak it. In the first quadrant, what I do is, okay, so this is like my thing, right? Like this. What I do is I take the loop, and a lot of them have like natural bends in them, which I kind of like, because what this allows you to do is that in the first quadrant, it allows you to like get a flat surface right here, so that way when you go to do it, like you see how my loop is like completely around it, you want to make sure to streak the entire quadrant. Some of y'all have just been doing like this part, but you want to like almost touch the edges, like come as close to the edges as possible. And then for the first quadrant, I do my inoculating loop really flat as well. Okay, and so that's your first quadrant. So then you replace the lid and you flame it. You flame your inoculating loop again, and then when I go to do the second quadrant, I take off my lid, and then I take my, and then for the first quadrant I do like it flat, but for the second one I do like the tip of them. And I know it's like not as sturdy, but the tip still like transfers bacteria. Like it's still an effective way to do it. And what I do is I go back and overlap the bacteria streaks I made in quadrant 1. I go over those 4 times. So I go 1, 2, 3, 4, and then after 4 I just take it and I just go down all the way to the bottom of the line. Alright, so then you put your lid back on, flame it, take your lid off, and what you do is your streaks that you made are oriented like this, so when you flip them, they're still oriented like this, and that way when you go into the next quadrant, you overlap perpendicularly the streaks that you made earlier. So still use the tip, go back, go back 4 times should still be good, and then you just do the same thing in the 4th quadrant. You flame it, or you put the lid back on, then you flame it, and then you do, again, rotate it to where you can get a perpendicular overlap, and then you go 4 times and then go to the bottom, and then you should be good with your isolating of the colony. Some of the problems is that y'all aren't effectively going overlapping, and that can be bad for isolating colonies. But if you, the point of the practical is to just get an isolated colony without contaminants. like I said, I think they take off for contaminants, and then if you don't isolate a colony, you don't get a good grade. But a lot of y'all are really good with this. If you're concerned or struggling, at the end of the experiment 6, like writing stuff before the worksheet, they have a common streak plate mistakes and suggestions page. It's on Milo Lab Manual 26, if it's remained the same, which I think it has. So page 26, and it goes through some stuff. So you want to make sure to do that, and make sure to be as careful as possible, be as clean as possible, be as pure as possible, and y'all should be and y'all should do well on y'all's practice.